tears. They convey happiness or sadness, but they do something else as well. In the early 1900s, a bacteriologist by the name of Alexander Fleming happened to create an accidental experiment. A teardrop fell into one of his bacteria cultures. It set the stage for a modern understanding of how cell processes are controlled. Many years later, the chemical in tears was isolated and described. It was an enzyme, now called Fleming's lysozyme. Lys means to break apart, zyme means enzyme. We now know how Fleming's lysozyme works. First, like all enzymes, it's a protein. A huge molecule built up from amino acid building blocks. Its chemical structure fits molecules in the bacterial cell wall like a lock and key, and once the enzyme locks on, the bacterium's cell wall comes apart. If you aren't in a crying mood, here is another enzymatic reaction you can easily study in class, or anywhere where abrasions are common and hydrogen peroxide is handy. You have probably done this, but did you ever wonder what actually was causing all those bubbles? Hydrogen peroxide is an extremely reactive chemical, guaranteed to disrupt delicate cell machinery. On the upside, it will kill many of the bacteria left in a wound. Our cells produce small amounts of hydrogen peroxide during metabolism, but they also provide an enzyme that instantly renders it harmless, the enzyme catalase. In this case, the enzyme attacks the hydrogen peroxide molecule, H2O2, and breaks off an oxygen, leaving good old harmless H2O along with some O. O's get together to form O2, oxygen gas. So now you know what the bubbles are. If you don't often get scrapes, try this. Lower a little cube of liver into a hydrogen peroxide solution. The glowing splint says it's definitely being given off. Eventually, all of the hydrogen peroxide has been converted to water and oxygen. But what has happened to the catalase? Fill up the flask with a new batch of hydrogen peroxide and off it goes again. Enzymes are not destroyed by doing their job. One catalase enzyme can probably break down millions of hydrogen peroxide molecules and keep right on functioning. However, if you want to deactivate the enzyme, just raise the temperature to around 50 degrees centigrade, and the enzyme action stops. What has happened is that the chemical bonds that hold the enzyme molecule in its proper shape for disassembling hydrogen peroxide have broken, and the misshapen enzyme can no longer function. The lock and key fit between enzymes and their target molecules is behind the thousands of chemical reactions that keep us alive. So let's dive into this and see how the process of building these catalytic proteins is accomplished inside these living cells.